This video is on heredity, so we have quite a bit to get into here. So that will include DNA and RNA, meiosis and mitosis, alleles, and included in that will be recessive and dominant traits, and then we'll get into Punnett squares and how to do those. So looks like we got 4 questions to consider as we're going through this video. The difference between phenotype and genotype. That's definitely important for your exam. What is the hereditary material in humans called? What must occur in order for a recessive trait to be present in offspring? And difference between meiosis and mitosis. Okay. All right. So DNA is the hereditary material in humans and almost all other organisms. So right away, we already got one of our questions knocked out right there, right? That second bullet point, the hereditary material in humans is called blank. There we go, DNA. Genes are factors that control traits. Genes are the units of hereditary and are a locus or region of DNA. They determine what traits are expressed in individuals. So being able to know in a couple sentences what genes are, that's important. Jumping back into DNA here, DNA controls the cell's production of proteins. DNA is shaped as a double helix, which looks like a twisted ladder, right? And so as you were looking at these two, that twisted ladder that we see over on the right side, right? That's DNA, that's the double helix, where the single ladder that we see on the left side is a representation of RNA. So each rung of the ladder is made up of a pair of nitrogen bases. There are 4 bases, and again, we're talking about DNA here, which are adenine, guanine, thymine, and cytosine. And they always pair with each other in specific ways. Adenine always pairs with thymine, guanine always pairs with cytosine. The sides of the ladder are made up of a pentose sugar and phosphate. This combination, a pentose sugar, a phosphate group, and the nitrogen base, makes a nucleotide. Okay, and so we can kind of jump into these. Nucleotides bind together in specific patterns to form the genetic code that defines a given trait. RNA is essential in various biological roles in coding, decoding, regulation, and expression of genes. In order for a protein to be expressed, it first needs to be transcribed by messenger RNA. This messenger RNA is translated into a protein by ribosomes. And then finally with codons, a codon codes for an amino acid. The amino acids are linked together as translation takes place. A group of 3 DNA letters makes up one codon. Okay. Meiosis and mitosis. We need to be able to recognize the difference between these two. Most of the body's cells are produced through mitosis, which is a process of cell replication that results in 2 identical daughter cells from a single parent. The number of chromosomes remains the same as the parent, which is 46. This is a diploid cell. Meiosis, on the other hand, results in 4 cells, with each containing a single copy of each chromosome, which is a haploid. Meiosis is a type of cell division that reduces the number of chromosomes in the parent cell by half and produces 4 gamete cells. This process is required to produce egg and sperm cells for sexual reproduction. Okay. So that's just something you need to be familiar with. One of our questions did focus in there on meiosis and mitosis and being able to recognize the difference in their types of cell division. Okay, as we jump into alleles. So alleles are different forms of a gene linked to specific characteristics. When 2 different alleles are inherited for a trait, but only one allele is expressed, that allele is called the dominant allele. The allele that is not expressed is recessive. If the alleles are alike, that person is homozygous for that particular gene. If the alleles are different, that person is heterozygous for that particular gene. And that should be pretty easy to remember, right? Homo meaning same, hetero meaning different. If both alleles are the dominant allele, the gene is called homozygous dominant. If both alleles are recessive, the gene is called homozygous recessive. If the alleles are different, it's heterozygous for the gene. Okay, so those are definitely important terms to know that you will be tested on. A recessive trait is expressed only if the offspring has 2 copies of the recessive allele. Okay, so that's important, and I believe we probably have a question on that. So that third bullet point there, what must occur in order for a recessive trait to be present in offspring? 2 copies of the recessive allele. Okay, so that's an important one that you definitely need to know. And then on the other hand, a dominant trait will be expressed even if only 1 recessive allele is present. Okay, so as we jump into phenotype versus genotype, which also I think is one of our questions, we'll go back and look at it in a second here. But knowing the difference, phenotype refers to a trait's physical appearance based on an organism's genetic code. Environmental factors can also play a role. But the genotype is the part of the genetic makeup of the cell, and therefore of an organism or individual, which determines a specific characteristic of that cell or of the organism individual. Okay, and so as they were asking us, most likely it's the question will be, what is the difference between a phenotype and a genotype, right? And so again, the phenotype refers to a trait's physical appearance. The genotype is the part of the genetic makeup of a cell. Okay, let's go back here. So that's the end of what we need to know for alleles. Okay, Punnett squares. The Punnett square is a square diagram, as you can see here, that is used to predict the genotypes of a particular cross or breeding experiment. It is used to predict the probability of the offspring having a particular genotype. To make a Punnett square, start by drawing a box that's divided into 4 equal squares. And you can see this on the screen, right? We have 4 equal squares. What we do next is label the rows with one parent's genotype and the columns with the other parent's genotype. After that, label each square, starting with the letter to the left of the square, followed by the letter above the square. And so you can see what we've done there, right? So in this case, we have, when we see those 2 capital G's, those are both dominant. We see those 2 lowercase g's, those are both recessive, right? And then, so we have 1 dominant and 1 recessive within each square in the Punnett square. So a classic example is parents who have, they both have brown eyes with heterozygous genotypes, right? And so when we say heterozygous genotypes, that would mean there's 1 dominant and 1 recessive, and so that would be expressed as capital B and then lowercase b. So in this case, we need to know that brown eyes are dominant over blue eyes, and usually the question would be, what is the probability that these 2 are going to have a blue-eyed child? So if you fill in the Punnett square, you'll get capital B, right? So 2 dominant, capital B, lowercase b, and then 2 lowercase b's, with the 2 lowercase b's being recessive. So the likelihood of getting those 2 recessive b's is 1 in 4, or 25%, and that's the chance that you have the blue-eyed child. Or as another example, say that you know that the father has curly hair and is homozygous dominant, right? So that would be if capital C, homozygous dominant. In such a case, no matter the genotype of the mother, his offspring will always have at least 1 dominant allele and thus will have curly hair. So this is to say that the probability of his having a curly haired child is 100%. You could easily see a question like that on the exam. Alternately, if an individual has blue eyes, which again is a recessive trait, we know that the father and mother each have at least one recessive allele, right? Okay. Whoops, too far. And then we can jump back into the questions, right? So based on everything that we've done, we should be able to answer these 4 questions. So this is a great time to hit that pause button, see if you can answer the questions. If you struggle with them, go back into the video because we've answered each of these 4 in this video.